some drugs will help in diagnosis. So drugs are of multiple types. So from where the drug design have been started. So the drug design is not a new field. Far enough, when the human civilization have started very long back, right from the caveman time, this drug discovery have been started. Correct. You might be heard of this. Like our ancestors, even they will go and they want to find a medicine for stomach pain. What they will do, they will create a stomach pain in monkey and they will tie the monkey for two days, and they will release in a particular time period, and the monkey will go and chase and find the right plant. So by the way, human have started discovering drug for a long time. But how about designing a drug? So why drug have to be designed? So all these aspects will deal today. And today we can design a drug based upon two perspectives like mind and machine. Correct. So we have a mix of both, like our human intelligence and artificial intelligence. So today I'll discuss about that perspective too. Right. So where it started? It's starting from making, it's starting from designing, and it's so AI intervening in the case of analysis too. So in all field, this drug design, AI is going to impact. Right. So we are very aware, right? We even the pharmacist will go on. You guys will go on have an experience in pharmacy field, and so let us have an interactive session, my dear participants. Like, what do you think the costliest drug you have sold, or what is the costliest drug you have recommended? Am I audible? So what's the price of paracetamol today? So rather than having a mono session, yes.
So the participants have been muted, sir, actually.
Oh, fine. It's not allowed. They're not allowed to speak, ma'am. Fine. If they are muted, thank you for that. Okay, you guys can also interact in meeting chat. Yes. So Dr. Baby ma'am is saying like 2 rupees per tablet. Correct. 1.5, 2 rupees. Fantastic.
Maybe less than 2 rupees.
Ma'am?
It is less than 2 rupees.
Yes, it's less than 2 rupees. Correct. Yeah, 2 rupees. Yes, Dr. Vijayanta ma'am is saying. Fine. So drugs are that cheap. Do you think all the drugs are that cheap?
No.
Not at all, right? See, the drug what I mentioned over here, like for example, paclitaxel I mentioned for 9,000 rupees. Correct? 9,000 rupees or 190 rupees, 3,600. Can you guess some costly drugs for the maximum price? What might be the cost you think? All of you.
Anti-cancer drugs.
10 rupees? Okay. Hey, look at the slide, please. Can you guess what kind of drugs these are? Like Herceptin, Avastin.
Methylprednisolone, more than 1,000 rupees for one dose.
Okay. So dear faculties, can you guess what the drug I have displayed? Like you can even use yes, correct. You can even use your browser and check what are these drugs. Hopefully you might be aware of it, but for a particular purpose, I'm saying like if you are not aware, you can very well check these drug names. Herceptin, correct. So Herceptin is a drug used mainly for treating breast cancer. Yeah, I got some answer from the chat. Good. So you either you can speak or as the muted, you can be active in the chat. So it will be a very live section for us. Correct. So Herceptin is for breast cancer. What about Avastin? Any guess? Anyone is searching? Or anyone is searching? Have you heard of?
Lower cholesterol, sir.
Yes, ma'am. Correct. Exactly. So bevacizumab. Yes. So MAB is none other than they are the monoclonal antibodies. So this kind of cancer drugs, fine. Again, it is prescribed for several types of cancer, okay, like ovarian cancer, kidney cancer, or colorectal cancer. For a lot amount of cancer have been prescribed. Same in the case of Erbitux, fine. So Erbitux is prescribed in Any answer for that? Again, it's a monoclonal antibody, correct? So where it is prescribed for? Yes, again, it is also prescribed for cell cancer, head and neck cancer. For multiple cancers, they have been prescribed for. So, my dear participants, being a pharmacist, why do you think these drugs are so costly? Of course, all the drugs are costly, but these drugs are a layer high costly. And do we think these drugs are going to cure the patients? And what's the cost of it? And we are suggesting them to take for 17 courses. So to roughly 75,000 into 17, correct? So definitely there'll be many lakhs, more than 10 lakhs, correct? So in such case, can you guess the reason? Yes, I'm getting some answers in the chat. Might be clinical trials. Due to clinical trials, they are getting costly. More than clinical trials, what might be the reason? Yes. Yeah, of course, clinical trials are so costly process. I agree with Dr. Ajit and Maria Sana. I agree, clinical trials. But we forget a main important thing. What is the fact? So there is a beautiful term called preclinical trials, or otherwise they are called as what? Drug discovery. Correct? So preclinical trials have been taking a long time. There are two reasons for all these costly drugs becomes costly. The reason is to discover those drugs is not an easy process. Discover that drugs are not an easy process. Why they are not easy process? Because they involve time, a lot of time. Nearly 14 years, like Ram went to Vanavash, like 14 years, that much time a drug will take to come out of the market. As you guys are more aware than me. And of course, yes. So of course, more than that, it's a tedious process. We can't pick one drug out of 5 drugs or 10 drugs. We have to pick out of a lakhs of drugs. Correct. So that's the reason it's getting so much delayed. For example, they may undergo a protocol called in vitro. Generally, in vitro refers to a test tube. Whatever the analysis taken in test tubes are in vivo in living cells. Either I can take in the case of cancer cells, or even I can induce cancer in the rats or rabbits. So I'm very fortunate enough to work in Madurai Kamaraj University, where they have applied for an ICMR project, Indian Council of Medical Research project. I'm basically a bioinformatician, and I'm very fortunate to work in animal models. So we induced skin cancer. We induced oral squamous cancer in the hamsters. We used to paint carcinogen in the cheeks, in the inner cheek, in the buccal cavity, and they will induce cancer. After cancers, we'll divide into several control groups. Okay, control groups, disease groups, based upon dosage levels. So then we have treated and we are just examined about their physiochemical changes, their histopathology, and so on. So it's a phase of preclinical trials. And of course, clinical trials is not also easy joke. They themselves will take 7 years. Correct. Starting with phase 1, phase 2, phase 3. Like tens, hundreds, and thousands of volunteers will be involved. And again, there is 6 to 7 years. And after the drug have been released to the market, even they have to undergo post-market monitoring after the follow-up of drug review and FDA approval. So this is the reason it is getting costly. And another important factor. What is the important factor? As I said previously, they are not only what? Time consuming. They are also tedious. So where do we start with? We start with nearly 10,000 compounds. We nearly start with 10,000 compounds. Then we moved on to 250 compounds and 5 compounds. Correct? So my dear fellow participants, a quick question for you. So what is the difference between PK and PED? Pharmacokinetics and pharmacodynamics? So I know it's a very, very basic question to the fellow professors, but I would be very happy if I get a quick answer or a smart answer from you. I myself type in chat PK and PD. And where drug discovery is mainly focused upon? PD or PK? Can we expand PK? So PK is none other than pharmacokinetics. Am I right?
Pharmacokinetics.
Fantastic. Pharmacokinetics. Correct. And PD is none other than pharmacodynamics. So exactly, pharmacodynamics. Let me put the phrase in this way. See, I have a knee pain. I have a knee pain and I am consuming some drugs. Okay. Right. What happens when I swallow the drug? Yes. Yes. I got an answer from Bangupali Jagadishwari. Yeah, she's true. What happens to the drug? The drug have to undergo lot of properties, right? Like admit, as you are very well aware, like absorption, distribution, metabolism, excretion, toxicity. So this is a PK wala, like pharmacokinetics wala. But once after overcoming all these challenges, once a drug reaches the knee, it goes undergo pharmacodynamics. What drug does to the body? Pharmacodynamics. So that's the actual scenario. So in the preclinical trials, we are more concerned about this pharmacodynamics. We are more concerned about this pharmacodynamics. Why we are more concerned about this pharmacodynamics? Because I will find, see, you pharmacist will find a way to go and deliver the drug, right? That is not my concern. My actual concern is the drug have to do a molecular mechanism. It have to really block. It have to be very targeted. See, even after selling cancer drugs of this much price, still the cancer patients are facing a lot of side effects, correct? What might be the reason? Because our drugs are not targeted. Yeah, adverse effects. Our drugs are not targeted because the normal cancer cells are normal cell is only proliferated into cancer cells. So the drugs are not able to differentiate between a self and non-self, like which is a normal cell, which is a cancer cell. So that's a reason we need the help of artificial intelligence over here. I am going to show you a slow transition, how it happened. So I'm being in this field for more than 15 years. More than 15 years. Of course, when I started my career as a student, I have seen drug discovery in a very small phase, but how rapidly it is getting grown up now. After the Nobel Prize have been given to AlphaFold, how rapidly it got grown up. I'll demonstrate that. I'll give a small glimpse of it. Fine. So stage 1 is about 10,000 compounds. Then moving on to the stage 2, we will go for preclinical development. And as I told about the ADMET PK. So can you evidently see this PK property over here? Like the effects on body, safety in humans, effectiveness of treating the diseases. Correct? So all flies under this category. And regulatory approval. And finally, we will end up in what? We will end up in one compound. That compound will be given to the market. Right. Yes. So let us quickly demonstrate one case study. Right. Can anyone guess the saint over here in the picture? How many of you are aware of Siddha system of medicine? Of course, most of you are from South India, I guess.
Yes, sir.
Yes.
Yes, sir.
Yeah. So most of you are from South India. Can you guess the saint? Any guess? So to have a traditional touch, just I included this slide. Okay, no one in the group. I can see some 150 people, 150 plus people.
Dhanvantri, sir?
No.
Dhanvantri?
Not Dhanvantri. Dhanvantri is from Ayurveda. Yes. Dr. Pooja have guessed. Yeah, Pooja and Dr. Baby have guessed. Correct. Dhanvantri is for Ayurveda, whereas Siddha started from Agastya. You are right. Agastya. Saint Agastya. And Kerala government have named, like even the Indian government have named the largest bio reserve in Agastya Biosphere Reserve. Okay. It's in Western Ghats. Again, Western Ghats is a part of Africa, you all know. So those who are not aware of it, you can go and check your school geography books, where when this Pangaea and Panthalassa, this Indian part, okay, have been separated from Africa and just clashed with the Asian continent, like Angara Gondwana. I don't want to go so deep, but you can check. Okay, so highly biodiverse. And same way in the case of Assam. In Assam, we have Western Himalayas. How biodiverse those thick forests, the same biodiverse we have in Western Ghats, starting from Maharashtra till the end of Nilgiris. Okay, even below the Nilgiris, it extending like Cardamom Hills. Okay, we have very rich biodiversity and lot of plant sources, no doubt in it. But additionally, I have seen people using pepper, clove. It's not even medicine; it's a part of our food, right? In biryani, none of the biryani misses these ingredients. Fine. Yes, correct. Dr. Bangupali is saying about the Gondwana spirit. So none of the food, okay, whenever we are making some non-vegetarian, definitely people will include this ingredients. Okay, I'm saying that these ingredients will cure cancer. Can I say taken for granted, can I say as it is? So what traditionally we'll do in lab, go crush the plant, crush the seed, extract the oil, correct? Or take out the compounds and go and treat in some animals, or go and treat in something. Is it fine? As a researcher, can we proceed like this? Not at all, right? As a researcher, we should not proceed like this. Rather, what we have to do? What is the first step you guys will do as a researcher? My dear faculties, I know you are all busy in academics, but of course, every faculty is a researcher themselves, himself or herself, correct? So, dear professors, what is the first step you will do to check whether this pepper or clove is having an activity? Fantastic. I got an answer from Mr. Ajit. Literature review. Yeah, Maria is also saying the same. What is the first step you have to do? Literature. So we should never forget this. My dear fellow faculties, like this world is rapidly growing. The moment today we are attending this FDP session in some part of South India, the same moment a Chinese professor might be publishing a paper regarding some plants, the same plants what we are discussing, correct? At the same time, an American professor might have deposited, a researcher might have deposited this structure in PubChem, correct? So no need to start from the scratch. That's what I always advise. So go to the literature and try to discover what have been reported before. And very painfully, lot of good plants from endemic to India is not reported properly. They end up in a very basic journal without a proper scientific evidence. They end up in very bugwash journals. They are not even having any impact factor. They are not even having any Scopus index. Of course, when you are not having a Scopus index journal, definitely your article won't be cited. Correct. So I always advise you to collaborate with some IIT professors or some university professors. Nothing wrong in it, right? To make your study even better. Yes. So my dear fellow faculties, let me come back to the story. So Agastya have mentioned that this pepper, this clove, this spices, okay, Kerala, the land of spices, Tamil Nadu border too. When we have spices, definitely those spices are going to have potent secondary metabolites. The potent secondary metabolites like caryophyllene. Okay. So through my literature review, I have found that the beta caryophyllene is a compound which also acts as a chemosensitizer and also involves in angiogenesis. Also involves in what? Angiogenesis. Clear? Okay. Here comes the question. Okay. So I got the pepper compound and I extracted the compound from it. Or I through GCMS, I found the compound name and extract the structure. So where can I imply that? Where do I have to test it? Someone is aware of these cell lines? What I have shown here, the left-hand side? Of course, the answer is there in the picture. But have you heard of all these terms like HCD116? No? Yes, colorectal cancer. Okay, so if not, you go and browse all these cell lines. So each and every cancer have cancer research have no standard cell lines are there available for that. So we can purchase the cell line. If any cell line laboratories are there, we can ask them to revive the colonies, cell line colonies, and we also can treat the drug over there. Okay, so if I found this answer, I want to test in different dosage, or I want to make a formulation, or I want to make a nano composite. So once I make it, I can purchase those cell lines. If I have a standard laboratory, then I can check the activity upon that. Correct? So the first case, the traditional knowledge should be supported by literature. The second case, I have to arrange all the equipments or materials for conducting the research. Yes. So how am I going to conduct those research? Look at my slide. So what is a regular protocol? Is there anyone from the audience who is doing this in the lab routine? Like how many of you are involved in extracting the plant resource, and how many of you are involved in animal models or cancer cell line? Even you are not able to text or reply, you can have a hand raise. So there is an option in Zoom where you can see hand raise. When you go to more, I think more or somewhere I have seen. Yeah, thumbs up symbol is there. Fine. Hand raise thumbs ups are there. So that I can have an idea like how many out of this total of 165 audience are aware of this studies. Only one. Okay, someone are involved in only in extraction, not about the cell line. Okay, thank you, team. Thank you. Yes. So again, some professors are involved in all those studies. So mostly are involved in extraction. Okay, thank you for the update. Fine. So what happens after extraction? So after extraction, generally we will go for, as I mentioned in the previous slides about clinical trials, preclinical trials. Either I go for in vivo or in vitro, correct? Of course, in test tubes or even in cell line. If I want to put in cell line, again, it becomes in vivo, right? So once it is done, I'll just evaluate the properties. But coming to the point, whether this whole study is required, whether I have to prove it, whether pepper is a very new plant. How many of you think already research have been done in pepper? Of course, many research should be done, right? Pepper is not belonging to us. Pepper is belonging to the whole world. Again, an interesting story. Do you know why Europeans came hunting India, Asia, Southeast Asia? They not came for money. They not came for gold. They came for pepper. They came for spices from India. Correct. There is even, if you go and look at your history books, there is even spice route. The same way they go and capture China for, they try to capture China for silk. So the spice is a well-known compound for the whole world. So of course, what might happen if it is well-known for the whole world? Not only Agastya who mentioned about the spice, of course, many knowledge might be already available. So in such case, I always advise you, no need to repeat all these steps. Rather, look at the slide. Rather, what we can do? Yes. What are the possibilities we can do? I know in plant compounds. If I know in plant compounds, what might be the alternative? Gomati ma'am, is the audience are allowed to speak?
Yes, sir, very much. They can unmute their mic and they can speak it.
Okay, ma'am.
Yeah.
Thank you so much.
Yes.
I know, see, it's a kind of peer learning, my dear fellow participants. It's not only about monotonous. If you are peer learning, you can share your idea for 1 or 2 minutes. That will be more. Yes, please. Yes, please.
Chemistry, artificial intelligence.
Sir, please. You're trying to say something? Yes. So my question is, if I don't want to extract the plants, where do I get the structures of compounds? Audience, even if you can't answer through unmuting your mic, you can also type. Where do I get the structures of my compounds?
Library, sir, from library, literature.
PubChem can give us the structures,
Fantastic. Fantastic.
and in silico studies can be done. That is what we do.
Yes. So is there only PubChem?
No, there are multiple natural sources, phytocompound sources, databases are there.
Yes, correct. PubChem, Drug Bank, Campbell, correct? Yes. See, there are very important things called primary databases, secondary databases. So, for example, UniProt. How many of you heard of UniProt? UniProt, NCBI, correct?
I have heard them.
Yes, yes. So if not, after the end of this lecture, you can go and search all these terms like UniProt, NCBI. NCBI is not only a database, it's a data warehouse where they have several collections of databases. There is a database for peptide, there is a database for nucleotides, so on and so. What do I try to say here is, this PubChem is also a part of this NCBI, right? Where they have deposited the entire information about the drug, particular drug. When I say pepper, it doesn't mean that only pepper. Inside the pepper, there might be several phytocompounds. Most prominently, eugenol. Have you heard of eugenol? The same eugenol might be present in other spices too. Okay, whatever might be the source, the compounds are so much important. So I can retrieve the structure of those compounds first. Same. Look at the right hand side. I don't want the animal to be injected with these drugs. Rather, what I can do? What is other possibilities? I mentioned about 3D structures of target receptors, right? So what might be the 3D structures? See, I want to treat colon cancer. Have you guys are aware of pathways, cancer pathways, apoptotic pathways? If any
Yes,
disease,
sir.
if any disease have to occur, what is the first instance we have to look is that to particularly cancer, there will be a block in lot of metabolic pathways. There will be a block in lot of metabolic pathways, correct? So when there is a block in metabolic pathways, what happens? We have to find the target receptors. So if I stop, it's like a cascading reaction. See, when you go to any parking area, okay, when you go to any parking area, just when you push one, okay, that is also called a lot of games are the Dorito games, I guess. When you push one cycle, it falls one by one, correct? The same way, when you stop one enzyme, it is a cascading reaction so that we can prevent the cancer. We can prevent the entire cancer. In such case, I have to find one target receptor. And the target receptors can be found in which database? Where do I get the structures of proteins?
RCBS.
Yes, RCBS is the
PDB.
yeah, correct. RCSB, PDB. Correct. If I do not have any structures in PDB, what should I do?
AlphaFold.
Correct. So AlphaFold is a kind of
AI tool, sir.
correct, ma'am. It's a kind of a modeling tool, AI-based modeling tool that is very advanced now. Why it is advanced? Because even they got Nobel Prize. Okay, 2024 Nobel Prizes for AlphaFold. It is very advanced because it have been trained with many number of protein structures and it predict what might be the conformation of each and every protein. So I can retrieve the structure over there. Even it is not there, there is a lot of modeling softwares. Okay, all around the world there is a community called modeling community. Models are not only like what cinema models are, persons who are displaying something. Models also mean a miniature of a real world, correct? So we can model those proteins. We can model those proteins. Yes. So once I model those proteins, I can simply compare. So this image is from one of the paper I have published recently, submitted recently and accepted for publication, where can you see the data? What happened over there? And I just checked with the protein called Aurora kinase, a protein. You can go and browse through this protein. Aurora kinase B. Okay, that I shortly call AKB. So what happened over here, my dear friends? I not only took one compound, right, beta caryophyllene, even though the research previously said about beta caryophyllene. Can you see here? In 2025, there is a report of beta caryophyllene. But in 2026, what I'm doing now, I not took only beta caryophyllene. Rather, I took their analogs. What are their analogs? Is the name visible? Alpha caryophyllene, caryophyllene oxide, isocaryophyllene. Correct. I took all these analogs. And tried to find their binding cavity, binding packet. Why binding packets are important? So when you take any enzyme, there is a thing called catalytic triad. Maybe 1 or 3 residues might be involved. So that is the actual key spot. When I want to break the lock, see, that is locked. If I missed my key, I can use a small hairpin to open the lock, correct? Same thing happened because I want to know the spot where I have to insert the hairpin. So this is the binding packet, key residues. So my compounds have bound in the key residues of that AKB. Okay, how do I evaluate among the compounds which one is best? Yes, many of you might have involved in CAD, correct? Computer-aided drug design. Can someone say which among them is very best?
First one, sir. Alpha caryophyllene.
Reason, ma'am? Why the first one?
Because the binding energy is more.
Yes, yeah, lower the binding energy. Yes. So in that perspective, binding energy is very lower. But unfortunately, there is another deviation. Compared to number of hydrophobic interactions, which one is higher? The third one, right? Can you see the third one?
Yes.
Isocaryophyllene. Even though their binding affinity is lower, their number of interactions are more. The third one. So this is not enough for me to prove whether alpha caryophyllene or beta caryophyllene. Then I have to move for another technique called molecular dynamics and simulation, where I set up the real environment of protein, water, salt, and then I'll simulate for a few nanoseconds. So this is a traditional, conventional way of doing CAD, computer-aided drug discovery. But where do AI involved over here? That's a question, and that we are going to analyze today. Right. So to answer the question, CAD is a magic bullet. This computer-aided drug discovery is what? A magic bullet. Why it called a magic bullet? Because it nearly saved how many years, my dear faculties? 2 to 5 years. Okay. And what is the cost of those drugs, that research? $500 million. So we are talking about the $1 million business. It can reduce time, reduce animal testing, improve success rate, improve the understanding of drug protein interactions. Am I correct? For example, from discovery phase, DAAC refers to discovery phase, from screening to lead compound. Okay, some 100 compounds we can reduce. And again, in the development phase, we can keep on checking in all phases and shortlisted 2 to 3 drugs. And finally, in the market will be 1 drug. So what if a company will appreciate you if you are saving $100 million? What might be your salary then? Even though we are not placed in high-fi drug companies, but we can start the prediction. We can start the prediction. Prediction is not only for our drug field. Prediction is existing everywhere. For example, today, is there a rain or not? Rain is not going to come or rain is going to come. Where do we know? Where do we know? How they are predicting today there will be rain? Yes, satellite data. Where do they predict? Satellite data, right? The same way, I can, if you give me 1,000 compounds, I can say not all thousands are going to work, right? And if you are giving me only one compound, I will inform the fellow companies or person who assigned me the work that more than this, there is a lot of variance. So what would be my result? I can cite their paper like so and so et al. Compared to your beta caryophyllene, I have found few more caryophyllene that is effective than your beta caryophyllene. Correct? This is the way drug is discovered. I have not taken any rat, I have not taken any drugs, but I have just finalized the results. And the labs who are having funding can go and test this. Clear? Yes. Yes, again, coming to this slide. How many of you are aware of all these different processes? Like LBDD, SBDD, system-based, correct? System-based drug discovery, ligand-based drug discovery, and structure-based drug discovery. There are several things are there, correct? Yes. So among the drug discovery, okay. So what we discussed so far is structure-based drug discovery, like molecular modeling, correct? Molecular modeling, molecular dynamics. Yes. What about ligand-based? Have you heard of QSAR-based, computational chemistry, molecular profiling? How many of you are aware of QSAR? And particularly ML-based QSAR? What is the full form of QSAR? I think my previous lecture was about that. My in the FDP.
Quantitative structure activity
Quantitative
relationship.
structure activity relationship.
Fantastic. So QSAR is a quantitative structure activity relationship. Again, this QSAR is very well coupled with AI. So the traditional QSAR is only done with only one algorithm. Okay, like a regression algorithm. But now, lot of regression algorithms. Now, lot of improvements have been came in classification, right? So these points we will discuss. Okay, today we'll discuss. Yes. So right from target discovery to clinical trials, there have been an entire set. Okay. So without going further details, simply if I know about my ligand, for example, in the previous slide, you might have seen about my ligands, right? I know these ligands, I know my protein. If both were, what kind of drug discovery you have to move on? Structure-based drug design. Whereas I know my ligand, but I'm unaware of my protein. I do not know where my protein lies, which is my protein. Then I have to go for ligand-based drug discovery, like QSAR, virtual screening. Clear? Where my ligand is unknown. I do not know what kind of ligand it is, but I might know my protein. Then you have to go for de novo virtual screening. Fine. Coming to library design, like high throughput screening, like HTS. Okay. Where none of them is unknown. I'm not aware, but I know only my disease condition. I have only research problem. Then I have to go for library design. Then I have to go for what? Library design. So this is the flow of CAD. So far, we have just a quick summary. So far, we have seen about what is drug, what is a drug design, correct? And why drugs are so costly, and why it is time consuming. Drugs are costly because of time consuming and tedious. And we have seen some case study. Okay. And instead of doing a traditional flow, we can also do an in silico flow, an alternative method. Clear? Any doubts in it so far? Yeah. Good. Thank you for your response. And we will have a small quiz. If time permits, we'll have a small quiz at the end of the session. Okay, once your discussion have been over, we'll have a small quiz. And let me check how many can able to answer. Yes. Yes. So this is about AML based CAD. So what is the difference, my dear friends? The previous Venn diagram I've shown and about this, what is the difference? Can you see a big difference? Yes? No? Yes. Like design and discovery. What's the, yes. What is the classical way and what is the key machine learning way?
See, we
Someone
have
try.
to put the input like omic studies, omics data, and descriptive data. We allow the machine to learn it.
Okay.
So that is the traditional CAD as well as AI/ML based. This is the difference.
Great.
So
Yes.
through the RAG, we'll retrieve all the information from the net or wherever the source will be, the open source will be there. It will get back and it will be agentic AI. It will be produced.
Oh, great. Okay, how far it is advanced than our existing CAD? So I have the answer, but before that, I want to know your awareness of this, dear professors. How far these machine learning methods, okay, like generative models or reinforcement learning, are advanced than the previous one?
It
Have
takes
you heard
time,
of?
but
Yes,
yeah,
sir. Yes, please.
it takes time when you're doing in each and every one algorithm will be there. When you're
Okay.
going to connect with all the open sources wherever it is there, even RAG as well as MPS will do this one.
Okay.
Wherever it will do and it will make a model for us. And Colab, it will do the coding and it will be there is a back end engineering and front end engineering it will be good, I think.
Correct. Correct. So
So your
partially it is.
large scale data can be analyzed and hidden molecules where our brain cannot process so many at a time, that will be coming up and analyzed by these softwares very fast with AI tools.
Okay. Good. So I'm getting a lot of new answers. Like you're saying about large data, correct?
Yes.
How about data? What's the difference between data and information?
I would say it will be coming in the form of a data. Different molecules that we will be screening, we will get definite parameters which we can analyze and measure them. So quantitative
Large,
and statistical evaluations can give us better prediction.
large
Oh.
information is data, sir. Small
Large
is
information
information.
data. Okay. You're putting in a reverse way. So thank you for all the answers. I'm very eager to know about your understanding of all these. Okay. Whether large information is a data or large data is an information. How can I put this one?
Large information is data.
Both ways, sir.
Large data is information.
Large data is information. Okay, great. Interesting again. Okay. I have answers for this. We'll hope. Then what is called knowledge then?
Understanding the data is knowledge, sir.
Understanding the data is knowledge. Okay. No, again, you're going back, very back. I would like to draw a pyramid for you. Right? Or I'll put in this way. When I have some random numbers, can you see my slide? My slide is visible?
Yes.
sir.
Yeah, just a minute. I'm trying for my pen. My pen is not working. Like, okay, I'm trying to write with my mouse itself. Okay, what I'm trying to write? Some 4 numbers I have written, correct?
Yeah.
And I'm writing some
Letters.
4 names.
Yeah.
Okay. Is there any relationship between them?
No.
No.
Yeah,
I'm saying some relationships are
numerical as well as descriptive data you're doing.
yeah, I have given some alphanumeric data. Alpha data and numeric data are given. I'm saying that these are the information I got from internet something. And I'm saying some meaning is there in the data. What are the possible meanings you can make out of this data?
Based on that, we can do the graphical representations, and it gives some information. Statistical basis, we can draw the graphical abstract.
Okay, no need to go that deep, sir. Just I'm asking, I have given
Maybe
4 numbers and 4 names. What might be there? What might
maybe
be
their age,
5
maybe
letters. So 5
Yes.
is the number, 5 letters, RAM, some coordination.
Yeah.
Like
Yeah,
We can
easy
have
guess might
different
be.
kind of permutation combination for all this data.
Great. Permutation combination. Good way of thinking. Okay. A easy guess might be what? The easy guess might be they are ages. They are ages. Correct? What might be the next guess? What might be the next guess? The next guess might be their marks.
Yes.
They are marks. Then I'm trying to understand some pattern over it. Is there any pattern about the marks?
It is in the multiples of 5.
It's the
Raised by 5 multiplied by 5.
multiples of 5. Is there any pattern in their names?
M and A are common in the
Correct. M and A are common. So my dear friends, that's it. That's about data science. When I gave 5, 10, 25, Ram, Sam, Ramya, Uma, these are all merely data. But what you try to do now is you retrieve an information out of the data. You retrieve an information out of the data. Getting my point?
Yes, sir.
So the 5, 10,
Yes, sir.
15, 20, 25 are called data. So my dear, my humble request, try to have a clarity of this in these keywords. We are using this word just for taken for granted. Like we are confusing with the word of information, we are confusing with the word of data. And today, because we are in a rapid evolving world of data science, correct? And we are saying about information technology. This IT field is there for more than 3 decades. But IT is something which make use of data. But today, the growth of big data, we want to know about what is data, what is information. Hope you now you are clear. The 5, 10, 25 is called as what? Just data. Now you derive a lot of information, might be their age, might be their marks, correct? That organized one is called as what? Is what? Information. And based on that, I can derive an insight. What kind of insights I can derive? I can make some business plans. I can make some what? Business plans. Or if I was a college admission in charge, I can derive a lot of information or insights out of it, correct? Right. That's it about. Coming to this machine learning part is something even a layer deeper than that. Okay, I know you have gone, you might have been expert in it or advanced in it, or a lot of bits of pieces of information scattered. So today's my job is to put in all this information into a consolidated story. Clear? Yes, and I'll come back to the slide. Definitely, I'll come back to the slide. Let us go and quickly have a glance about what is called information, what is called data, and I'll come back to this. Then I'll say why machine learning or discovery is very important in our drug field. Yes. Coming to data everywhere, correct? It's a real thing, my dear friends. Like the minute when we are explaining what is data, how many data is generated in internet? Is my screen visible?
Yes, sir.
Yeah. So how many of you are having social accounts? More than 3? Can I have a quick hand raise? Social accounts in the case, WhatsApp, Facebook, Twitter, YouTube. Out of 172 participants, yeah. If you want to share your number of social media, yeah, also fine. Great. 3, 4, great. Some people are having one only, yeah, great. Yes. So you have multiple thing, right? So each and every day, Okay, thank you for your response, everyone. Each and every day, how much information we are posting to the community, right? The moment I'm speaking, 300 hours of new video might have uploaded. Yes, 77,000 Netflix users have been using. Snapchat, Tinder, Uber, 694 Uber bookings might have happened in this one minute. Okay. So who is going to make use of this data? It's not just numbers, my dear friends. It's a business, billion-dollar business. Correct?
Social media, maybe marketing persons.
Marketing persons are using. All the producers are using. My question is, these big data are available and people are making use of those big data. What about the big data in biology? So where do we generate data?
Genetic information.
So starting from a very macro scale, the first macro scale is health records. See, to my knowledge, I have seen some Indian students. They are easily placed in some 20,000 rupees job or 25,000 rupees job. If he is a life science student, what is the immediate job for him? Medical coding, correct?
Yes.
So what medical coding people are doing is none other than they are maintaining the insurance records, health records. A patient, what's the age, what's his scenario. All this health records is also big data. What are the medications have advised to them? The insurance companies is also big data. Social data and behavioral data, psychology people, correct? What are psychology people are storing? What is his behavior patterns? This is also data. And now government is Indian government is compelling for us to undergo One Health. Do you know how many of you are aware of this term called One Health, my dear fellow participants? What is One Health? I'm really sorry, I forgot my pen. I'm trying to write with my mouse. What is One Health? Okay, One Health is none other than when I draw a pyramid. Okay, when I draw a pyramid, I'm putting like cockroaches.
Our health, environment, and social awareness totally called One Health.
Fantastic, fantastic. Correct. So just thank you for that. I'll just finish this. So I'm putting all plants, all animals, okay, and putting human as the top. Okay, this is called ego. This is called as what? Do you think we are the topmost of all organisms? Never. One Health is something we are part of it. We are part of other, like we are part of other plants. We are part of the society. We are part of this environment. Correct. So as the ma'am who informed that is none other than One Health is a combination of environmental health, animal health, and human health. So in such case, from environmental data, we are getting a lot of big data for biomedicine. Where do you dispose your chemicals? Where do you dispose all these stuffs? Clear? That is big data. What about transcriptomics? Yes, make it fast. What is transcription? Your basic molecular biology. What is transcription? DNA to RNA, RNA to protein?
DNA to mRNA.
That is called as what?
Transcription.
Transferring one form to another form.
And
correct.
protein synthesis is translation.
Translation, fantastic. So when I say transcription, that is not a single process. Lot of heterogeneous RNA are involved. When I have the whole set of data, it becomes omics data. It becomes omics data. And similarly, what is called metabolomics? Same way, when I say about a lot of secondary metabolites, it becomes metabolomics. So those data, and again, genomics data, omics data, correct? So all this data become a big data in biology. Like just couple of minutes we have spoken about the data in the social media, correct? Data in the social media, and people are making money out of this data, correct? And these data are available free in internet. We being a biologist, we being a pharmacist, how we are making money out of it, or how we are using for our research and thing. These data are freely available. These much data are there. Does any one of us are trying to take an information out of it, or retrieving a knowledge out of it, and make some predictions? If not, take it today as an eye opener for doing that. Okay, go and browse all these stuffs. I'm taking a privilege of saying this. Go and browse all these omics databases. We have millions of informations updated. Data are there. We can retrieve the information out of it and devise some work plans. Clear? Okay. By that, what kind of impacts we can make? Yes, drug repurposing. So can quickly say someone, we are very evident of this drug repurposing. Can someone say an example? I'll give a clue. This drug repurposing is a successful one during COVID time.
Aspirin.
Aspirin, ma'am? No, more than that. During
COVID?
In COVID,
That, yeah,
Dolo,
antiviral.
Dolo 650? Paracetamol.
No, ma'am. No. Very, very famous. There was a demand for the drug too.
Doxycycline, sir.
No, sir. No. Okay, I'll give another clue. It's anti-malarial.
Chloroquine.
Fantastic. What kind of chloroquine?
Hydroxychloroquine.
Yes, ma'am. Hydroxychloroquine. Correct. Yes, ma'am. So why I'm saying all this hydroxychloroquine, people came to know it is not only binding with the malarial protein, it also can bind with the COVID protein, like spike protein. It's all predictions again. I have given my students are working on this aspect. Their job is to retrieve the data sets from whatever the servers available. Like I can use the antibacterial drugs for anti-cancer. I can check for anti-parasitic drugs for anti-diabetic. Anything is possible. So those who are listening, us, if you're interested in this kind of research, you can very well approach us. Okay, you can very well approach us, and we can do a potential collaboration for this. Correct. Yes. Apart from that, accessibility healthcare, improved clinical practice, improved diagnostics, efficient community medicine, everything is possible in this big data. Clear? Yes. Any doubts so far?
No, sir.
Yeah. Hope you guys are along with me and enjoying.
Yes. It's clear. It's clear.
Thank you so much. Thank you so much.
Yes, sir.
So my objective is not to deliver the answer. My objective is to make you think a lot of questions. So at the end of lecture, I want you to ask me many questions so that I'm happy that you got the point. Okay. So we have n number of data, correct? In big data, we have n number of data. Just now we have seen these data. How we can make use of the data? What are the equipments or what are the tools I need to make use of this data is none other than you want to know maths, you want to know statistics, you want to know programming, you want to know machine learning. Sir, I hate maths. How can I go and do that? No need to hate math. You can have a collaborator with mathematics. You can take a collaboration with the programming people. And machine learning is for all. Anyone can master machine learning. It's not a tough task at all. To drive the car, you want to be a mechanic? Not at all, right? To drive the car, you not at all want to be a mechanic. You want to know how the car functions. That's enough. The same way in the case of machine learning. But you want to create a car, you want to know all these stuffs like maths, statistics, and programming. So if you know that, you can make an actionable insights from all this structured and unstructured data. In today's scenario, we are filled with a lot of data. We want to make an organization out of the data. That's it. Yes. Now comes another interesting story, my dear friends. Like human intelligence and artificial intelligence. How many of you think we humans are more intelligent than any machines have created in this world?
No, human being is more intelligent than technology.
Technology. So true, no, ma'am. Like we only created the technology, correct? But if I ask you 5 into 5, you can answer easily, correct? 25. When I ask you 555 into 255, what you will do, ma'am? We'll struggle. Even me, anyone will struggle, correct? We are not that much good in counting. But we can clearly say this is orange color, this is orange color, and this is color is blue. Am I right, ma'am?
Yes.
Because we humans are good in qualitative.
Yeah.
We are good in what?
Quantitative.
Qualitative. Quality. Computers are good in quantity. The very word compute refers to count. See, in Tamil they call kanini. It's miss math. Count. Compute means count. Correct?
Yes.
Yes. In such case, AI is trying to mimic the humans. AI is trying to mimic the humans to make a decision making. Not only counting, we have several skills like intrapersonal skills, linguistic skills, logical skills, spatial skills, musical skills, kinesthetic skills. These are skills of human intelligence. Someone is excelling in that field and they are trying to shine in their life, right? But how smart is AI, you know? This AI is getting trained out of it, and AI kept learning. This is the AI one we are encountering daily. Of course, I know everyone are having AI in your system because you are having email, right? When you have email, this very spam filter is an AI. That is called support vector machine. Like it's trying to differentiate between good mail and bad mail, correct? But a human will take a lot of time to understand, and it's a tough task for him. But AI can easily learn, reason, problem solve, and decision make. Clear? Am I making it?
Yes.
Yeah. Yes. Now it's a time for us to understand the difference interlink between data, AI, and machine learning. To be simple, I have a slide for it. Yeah. Can you see here, everyone?
Yes.
Machine learning is what? A subset of AI. Machine learning is a subset of AI. And data science is not a part of AI, whereas data science is a separate domain. Okay. But it integrates with AI and ML. It integrates with AI and ML. That's why we have to have a clarity. What is data science? What is AI ML? Now, if you go to any engineering college, there is another famous course where people are highly competing for learning this. Yates. You know that? So people are offering engineering in Yates. AI and data
Artificial
science.
intelligence, data science.
Data science.
Yeah, correct. When I ask I belong to a university where we have engineering campus, pharmacy, physiotherapy, and even law firm have been started. They will say 8 student. Okay, they are not aware of this term. Fine, whatever may be. Like look at the right hand side. What I have shown you. Label data, unlabeled data. Correct. So this kind of data is called supervised learning. Like I'm instructing the program what is carrot, what is tomato, and what is a capsicum. And it will make bell pepper in Western world. Like it makes a proper decision. Okay, where I'm not given a test set data, I'm not defining what is it, and my machine is able to detect it. This is a kind of supervised learning. The same in the case of what? Drugs too. Yeah, let me go back and come here. Yes. It's very, very important to understand here. Listen carefully. Like data science gives data, correct? AI learns. Once AI got learned, what it will do? It's like a loop. It generates again another data. And finally, we have a data, we have an AI, and we have the patterns learn. And which simplifies our job. It simplifies our job. These are the things we have seen. Yeah. Can anyone make sense out of this particular image? This is from a review paper. What happened over here? Am I boring you all?
No, sir.
Absolutely not, sir.
No,
Yeah,
sir.
thank you for this. Yes.
It's like a smile, and then the string, there is a numerical, and there
Yes,
is a code.
great. So, okay, the left hand side is the structures. I agree with that. Can you guess what are these?
Language.
Might be
Computer language.
carbon, one is
Computer.
carbon, like
Computer language.
Computer language, sir.
coding.
Yeah, computer language, some binary data. Okay, I'll put the right word. It is called descriptors. They are called as what? Descriptors. Okay, so what is descriptor again? The very, very important concept. When you understand descriptors, you can understand the entire QSIR, you can understand the entire machine learning models. Okay.
It describes the properties of the molecules.
Very good. Describe the properties of molecules in what terms? Okay, let me ask a very important question, then I'll jump into the answer. My questions are very basics. Which one is called as mother of science? Whether science has any mother?
No.
Okay, like it's a what do we?
From atom.
Yeah, it's a philosophical one. Like we have several sciences, right? Biological science, physical science, chemical science, geographical science, data science. Among compared to all sciences, which one is the basic science?
There is no comparison.
Ah. But which one should be the very starting point, ma'am?
Biological.
No, ma'am. Math.
Maths. Okay.
You can confidently say, you can refer anywhere. Maths is the mother of science. Math is the mother of science. In today's scenario, if you are ignoring math, you can't do a proper science. And that too, particularly in AADS scenario, like artificial intelligence and data science scenario, you want to do a basic math. I don't want you to take your notebook or don't want to go and memorize the periodic, this what table they will say? Multiplication table. That's not math. We want to understand logically, we want to make decisions. For example, the very word descriptors. I do not know whether I have that slide. I'll show. I'll check for. Yeah, look at here. This is the thing. So I said a few minutes back, like we are good in confirming colors, but computers are good in counting numbers, correct?
Yes.
Yeah, I have a computer and I want to make the computer to work for me. So what can I do? I have a set of, I'm a chemist or I'm a pharmacist. I have a set of drugs with me. The first thing what I'll do, I'll convert all these drugs into some numbers. How it is possible? What are the numbers I'll say?
Smiles.
Yes, yes.
Smiles.
Ma'am, smiles are not numbers, no, ma'am. Like smiles is a notation.
Okay.
Yeah. Rather, can I say the molecular weight?
S.
Okay,
They are numbers, right?
right.
Can I say number of hydrogen bonds they have?
Yeah,
These are again numbers. When I go on some advanced level, I will say the surface area. I will say about their volume. So for a particular molecule, I have now several numbers for it. These numbers are called descriptors. These numbers are called as what?
descriptors.
Descriptors. These are descriptors actually. So these descriptors I will have and try to make a lot of decisions out of it.
Hydrogen.
Yes. Can you see here? Like
Hydrogen.
molecular weight is a 1D descriptor. Whereas 2D descriptors are topological surface area, rotatable bonds, correct? H bond donors, H bond
Yes.
acceptors.
Yes, sir. Yes, sir.
And 3D descriptors are none other than polar surface area, molecular surface area, molecular volume.
Molecular volume. Yes.
Yeah, these are all descriptors. So I make my computer to work for me by converting those structures into numbers. Clear?
Yes.
And these are called algorithms. I'll develop an algorithm. So my dear friend, man is none other than without his tools. We are nowhere if you do not have any tools. Tools starting from the wheel, fire, and till date we have computers, a powerful tool with us. With these tools, if you use effectively, you become a champion out of this world. Like you can compete among anyone. Right? That's why today's kids, Gen Z kids, are earning millions by sitting in home. You know, they just do something in computers. They are earning in lakhs. But we, being a professionals, we know drugs. We can use the drugs to convert into numbers and try to find their activity. Yes. And again, before coming to the slide, again, I'll go back to a very important thing. Yeah, I said about, just now I said about numbers, right? Descriptors. So all these descriptors evolved from dimensions. So 0D, 1D, 2D, 3D, 4D. Clear? So those
Yes.
dimensions, only I converted into descriptors. So you don't want to fear about math. You want to know what is the logic behind it. That's it. So this is a mathematical formula, right? But whether it is tough to understand. So I'm a very average student when I was in school. So they made me to fear about math. But when I came to college and when I started to teach, I found math so interesting than any other science. Yeah. Length, weight, width, and height, that becomes volume, and that volume is a 3D descriptor. Whereas this one is 4D descriptor. Tesseract is a 4D descriptor. Clear? Yes.
Yes.
Yeah, and what else? Yeah, coming to this again, interesting story. I know ML. This ML based QSR generally function as about with a known activity and unknown activity we can predict. For example, I have a group of people. Among them, someone are healthy, someone are diseased. I want to differentiate them. Then I can calculate their parameters and make one small statistics. Then it gives us which group of people are healthy and which group of people are diseased. Clear? Yeah. Based upon their physicochemical properties. And if time permits, we also plan for a workshop like how to conduct all this, how to convert these numbers into how to convert the chemical properties into numbers. So a lot of opportunities are there. We'll discuss about that in future classes. Right? Yeah. And about regression and classification, very, very important point over here. Like, okay. Do you know the climate of Ooty? The JSS college is conducting a meeting today, but unfortunately we are in online meeting. If it is an offline meeting, what would be the climate of Ooty now?
Cool region, no?
Cool region, correct. Okay. So ma'am, when you say cool, you belong to classification. You belong to?
Okay.
Because you say it's cold or hot, correct?
Yes.
Gomati, ma'am, what's the temperature over there in your place? I think ma'am is not listening.
Sir, now around 20, sir, 15 to 20 degree.
Thank you, ma'am. Thank you. See, they are enjoying life. So 15 to 20 degree there. When I say this one, it becomes what?
Regression.
Regression. So I have to train my machine learning in either way, either classification way or regression way. Okay, here it is categorical and here it is continuous.
Yes.
Just now it's shown you the diagram of supervised. Both can be supervised.
Okay.
So when I build a machine learning model, either I have to go as a category, I have to go with the values.
Values. Okay.
Yes, ma'am. So the one value is, for example, I'm working in a cancer drugs. So generally I'm a cancer cancer bioinformatician where I work in drugs. I'm preparing drugs for anti-cancer. Okay. Where I derive the information of this IC50 from PubChem database. Okay, ma'am. Like, what is IC50 again? Anyone from the team? Audience? What is IC50? The answer is given there. IC stands for inhibitory concentration. IC stands for what?
Inhibitory concentration.
When I give 50, it refers to
50%.
50%. Correct. So when I say IC50 in numbers, okay, I'm saying 3 μg per ml, then it becomes regression.
Okay.
When I say I have some threshold value, like above 5 μg per ml, it becomes, what I mean is, when I give a drug of this dosage, the cancer cell will cleave.
Okay.
When I give a drug of that dosage, if I'm feeding the mechanism, for example, for a particular drug, what we told, bevacizumab, right? For that drug, we have this IC50. If it is proven in the lab, I'm looking for some novel inhibitors. Okay. Then I can come, instead of going for an experiment, I can conduct a prediction and compare with their values. Okay, this can be very well understood when only during workshop time. And this you can now assume and you can go and refer to theory. Clear all?
Yes.
Yeah. And that's a limitation of today's session, but hope I can started you to think about this aspect perspective, I guess.
Yes, sir.
Yeah. Okay, now let us go for a fancy names. KNN, LR, naive Bayes, random forest, support vector machine. How many of you are aware of all these fancy names? No one? At least in research articles?
Yeah.
Like these are none other than, they are machine learning methods. Okay, like naive Bayes, random forest, support vector machine, and particularly ANN, artificial neural networks, are more similar to our human brain. Okay, like how do they make decisions? All these stuffs we can say later. Yeah, that's what I design my model, machine learning model. Once I design the machine learning model, what I do, I'll conduct a performance and how far my new drug is good.
Okay.
Fine.
Yes.
Yeah. Then I'll make a statistical calculation and I'll try to identify how our drug, what kind of, I can't take the IC50 as it is. I want to convert into a negative logarithm that I call as PIC50 to reduce the dimensionality. I will reduce the dimensionality and as a result, I will look at the R square value. Yes. So by doing all this ML based QSAR, I can again going for reducing the animal testing. I'm getting a cleaner chemical. Okay. A lot of easy ways are there. Yes. Here comes the ML part of it. I have a diverse structures. And I train my machine learning model. On what basics I am training? I'm giving a lot of descriptors to it. Okay. By looking at the descriptors, my machine will now learn and take its decision by itself. Correct?
Yes.
By once, for example, you are the supervisor and you know everything, and you are training your PhD student, definitely that PhD scholar will train after his second year or third year, he will train some M.Pharm students, correct? Master graduates. Because being a supervisor, you have trained him. The same way, I have trained my system. The system hereafter doesn't require my inputs. It can decide by itself. How it can decide by itself? I'll compare my training set with the test set. And I'm looking like my test set are very well oriented towards the regression line.
Okay.
And they are having a good R square. R square above 90 is always good. Then I said my model is good enough. Then I can make a software. I can publish this. I can claim a patent for my model. Yes. Yeah. So orienting towards the topic. What does the word next generation refers to? Because our topic is more than AIML, Anand sir has given me a very important task of covering next generation. So what are the next generation, my dear faculty members? What is first generation, second generation, third generation, and what is next generation?
Next generation is advanced generation. They are must and should equipped with AI knowledge.
Correct. What happened in first generation, second generation? Why it is more advanced than anything else?
In first generation, there is manual power is more. They are searching something. They will take more time for invention something.
Correct, ma'am. More than that, they are less informed. They are
less right.
They do not have that much information to take a decision.
Right.
How many of you really appreciate your chat GPT? Can I have a quick look? Like how many of you are really enjoying your AI world? Only two? I would be happy if some who is listening today, really you feel that your chat GPT or your Google filter is helping you, you can give a hand raise. There's an option called hand raise. If you see the more button or chat button, there is an option called hand raise. Yes. If not, if you are not used to it, I kindly advise please use it. Because I can roughly divide, like scientists, people are roughly dividing this whole world into before AI and after AI. We used to say, right, before Christ and after his death, Anno Domini. Same like there was a huge revolution before AI, that is called electricity. So people divided the world before electricity and after electricity. Correct? Imagine the world without electricity. We 175 people are connected towards each other today because of electricity. Am I right? The same revolution what electricity did to the world, the same kind of revolution is going to be done by AI. So please start working on AI. Correct? Okay. And look at here. Like a massive shift in medicine from chronic symptoms to permanently repairing, curing biological diseases at their source. We don't want to treat in a different way. No need for surgery, no need for anything. We are very well informed in the sense we can create a drug and we can focus towards it. Okay, in animal modeling and testing, AI is doing a revolution. And AI helps in better selection. How it is helping in better selection? How it is helping in better selection?
Based on input.
Comparing.
Data will be scrutinized and then sent to us.
Sorry for that. I have a small issue. Can you come again, ma'am?
Based on our input, that give the best option.
Comparing.
Yes. Yeah. See, just what I said, like drug selected, whatever the existing drug, for example, 100 papers published in internet, like what drug, what kind of nano carrier they have used. Drug and carrier. Okay. When I have 100 data, I can use that data for building my own drug, correct?
Yes.
So it's all called informed decisions. It's all called informed decision. Informed decision. This decision is very, very important because you are well informed. You have to train a model. Training a model is not a big deal. You want to learn, everyone who is in the audience today, my humble suggestion, go and join some Python course. So dear audience, are you aware of this language, Python language?
Yes, sir.
So this FDP, you can take it as an eye opener. It is not just hearing the class and just skipping it away and getting a certificate. That is not there. If you want to survive in this next 1 or 2 decades, you want to enter into AI. And of course, if you are ignoring AI, that's well and good. It's your life, you can lead your life, nothing wrong. But professionally, if you want to succeed, my humble suggestion, learn Python. Through Python, you can perform AI. If you don't want to use Python, you will be an AI user, you won't be an AI creator. Clear? Yes. Okay. And let me complete this slide. How far AI is helping in nano system? Like they help in patient demand and routine therapy. Using patient demand, you can create a new nano system. Same way, AI in next generation therapies will help in selecting the drug repurposing or drug reposing. How we have to reassemble the drugs based upon the effective simulation. Based upon the effective simulation. So to answering my initial question, I have asked you a question, no sir, like how far I'm going to find a cancer drug. Correct? So ancient therapy will stop over here. We can't go for precision oncology. Whereas, and this is ancient. This is current allopathy. Like this is Ayurveda, Ayush, you know Ayush?
Yeah.
Yeah. So that methods people are trying to intervene. And these are our allopathy. People are trying to intervene using modern therapeutic interventions. But still no, there is no cure for cancer. Only we are trying to extend their lifespan.
Yes.
Or we are giving additional support to, that's it. Okay. But in near future, if we are enabling the AI integrated diagnostics, AI integrated therapies, I'll go back to my previous very first slide. This price will definitely fall down. We can make an affordable drug. We can make an affordable drug for 75 rupees even. Okay. This might happen in next 10 to 5 years, 5 to 10 years. If you achieve this, definitely the whole world is going to thank you, because cancer is not an incurable disease. Cancer is an unaffordable disease to cure. Correct?
Yes.
Many lot of deaths are not reported because they are not aware even they got cancer or something.
Right.
So one single cigarette can has having 75 carcinogens. I always advise my student, don't stand before a smoker. If your friend is a smoker, avoid him because he is supplying you cancer carcinogen. Okay, so with this take-home message of AI is very, very important for your next generation therapeutics, I end up my discussion here. Thank you for your patient hearing. Thank you all. Any quick doubts?
Sir, for talking, which like how we can get to know which drug is the best for the like activity? Like as you have shown one of the
I'm really sorry that your voice is not that audible. Can you please keep your mic closer? It's from my side or their side?
Sir, it's raining over here. Might be the network issue.
Ma'am, let me go one by
Is
one.
it audible?
Yes, ma'am. Yes, ma'am. Please, ma'am. Is it Maria, ma'am?
Yes,
Or
sir.
Dr. Debbie, ma'am?
Yes, sir.
Yes, Maria, ma'am. Yes, now you are audible.
Sir, so for docking, as you have one of energy will be best for the active site. So
Okay.
question is like, is it the least value? Also, like what I got to know is it the best value or best ligand site will be between the range of -7 to 11, the binding energy site for from docking.
Ma'am, that is not the only deciding factor. From after docking, yeah, you have to also go for non-covalent interactions, ma'am. Like hydrogen bonds, hydrophobic interactions, salt bridges. So based on that, you have to take a decision. And as I shown in my slides, even then you can't take a decision, proceed for molecular dynamics.
But the software is having the charges also. Like
No,
it is
ma'am,
very
no.
costly.
Again, it's a great myth. People are saying that, again, people are charging a lot. For example, Schrodinger. Don't go for commercial softwares. If you go for, I'll suggest to the entire group, there is a thing called WSL, you know? I am putting in the chat. People can follow that. WSL stands for Windows Subsystem for Linux.
Okay, Linux.
Yeah, the thing is, you can install Linux, ma'am. Until you guys install Linux, you are not going to make use of the Python, the AI, and so on stuffs. So please install WSL. It's a matter of 5 minutes. And lectures are available in YouTube. You can find the YouTube lectures on how to use Ubuntu. You can start reading about that, and no need for pay anything. You have a lot of free tools in Ubuntu, Linux.
So, sir, we can proceed with the molecular dynamics also.
Fantastic.
The WSL.
Yes. So I'm giving my mail ID. If you have any specific doubts, you can approach me.
Yes, sir. We can ask.
Yeah, you can ask the organizer and you can approach me if you have any specific doubts. I'm ready to clarify.
Okay. So, sure,
Yeah.
sir.
Thank you so much. Any doubts regarding today's session? Or any general doubts also welcome.
Sorry, in nowadays in market, so many books are available on basis of AI in pharmaceutical sciences, AI in some pharmacology.
Yes,
Is
ma'am.
that possible in pharmacognosy and HTT?
You want to imply AI in pharmacognosy, ma'am? Like that?
Yes,
That's your
yes, sir.
question?
Yes, sir. Yes.
Ma'am, AI is admitting wherever there is data. When you have some of data, definitely AI can be make use of it. But I'm not aware of the exact softwares. Let me check and say, ma'am.
Oh, okay,
But
sir.
regarding any AI tool, I advise you to start with Python.
Okay, sir.
Python is the starting point.
I'm trying to write one book basis on that, but I'm still thinking, is that possible in cognosy and herbal drug technology? That's why.
Very well possible, ma'am. It's possible.
Okay,
But
sir.
please collect relevant data from literatures, like scientific
Yeah,
literatures.
sure, sir. Sure.
Yes, ma'am.
Thank you so much, sir.
Most welcome. I think you are a very active participant today.
Yeah, I'm very much interested on this, sir.
Thank you so much. And thank you all for your wishes in chat. I'm looking at the chat. That hope these wishes are from heart. Like you really understood today's scenario and understood my lecture.
Thank you, sir.
Yes, ma'am. Thank you so much.
As we come to the end of today's session, I would like to express our sincere gratitude to Dr. Chandrasekhar Punnasamy, sir, for sharing his invaluable knowledge and expertise on artificial intelligence-powered drug design, from computational intelligence to next-generation therapeutics. Your informative, interactive session with insightful presentation has enriched our understanding of the transformative role of artificial intelligence in accelerating drug discovery and shaping the future of healthcare. I also extend my heartfelt thanks to the respected organizers, the Center for Advanced Molecular Design and Simulation, JSS College of Pharmacy, for organizing this faculty development program. And also, finally, I thank all the faculty members, research scholars, and the participants for your enthusiastic participation and active engagement throughout the session. I hope today's lecture has inspired all of us to explore the exciting opportunities that artificial intelligence has offered in pharmaceutical research and drug discovery. Once again, thank you all for your presence and participation. Have a wonderful day ahead. Thank you, sir.
And Ms. Harda, I request all the participants and the speaker to open their videos for the group photo. Harda, can you take a group photo?
Yes, ma'am. Sure, ma'am.
The participants are requested to just open their videos.
Madam host has disabled video. Can you
Yes,
please
thank you.
enable?
Thank you so much. Thank you, all the participants, and thank you, sir, for your wonderful and fruitful lecture session.
Thank you so much, ma'am.
Yes, sir.
My pleasure. My pleasure.
Thank you so much, sir. Thank you so much. And Dr. Duryanan, sir, will communicate to you, sir. Thank you so much. We all
Ma'am.
can leave now, sir.
Thank you all. Thank you all.